Let's talk about proteins. For most of us, they're a word on a nutrition label, or something that makes a terrible-looking meal sound tolerable. But proteins are so much more. They're the gritty, uncelebrated workers of our bodies, working away to keep us in one piece. There's collagen for skin and bones, keratin for hair and nails, amylase to break down starches, hemoglobin to transport oxygen, ferritin to store iron, myosin to contract muscles. Over 100,000 proteins are hard at work in our bodies 24/7, quietly doing their thing. Until, of course, something goes wrong. In this episode, we'll wade into the world of proteomics, the study of proteins that aims to figure out what they do, how they interact, and what role they play in health and disease. I'm Silvia Lazzaris, and this is The Science of You, a podcast series about the groundbreaking science that's changing the way we approach healthcare. Together with those who are leading the charge, we will journey from the discoveries that transformed our understanding of the human body to the innovations that promise to revolutionize treatments and medical practices. The Science of You is a Cora Media podcast, supported by the European Partnership for Personalised Medicine, coordinated by Fondazione Toscana Life Sciences, and co-funded by the European Union's Horizon Europe Research and Innovation Programme.
Proteins are the building blocks for building most of the components of our body. They're the workers that are carrying out most of the functions day to day for most of our organs and most of our biological and physiological systems. The best way to think about it is to probably think of the body as something like a factory. So DNA is the parts list. It's the list of all the raw materials and the components that are available for production. Proteins are important because at the end of the day, the actual biological layer that's influencing and causing disease is usually proteins. This is also why a lot of therapeutics and drugs that are developed to treat disease also target proteins themselves.
This is Austin Argentieri, population health researcher focused on human aging at Harvard University and the Broad Institute. We might be used to thinking about protein as a macronutrient in our food. But what's the difference between the protein in food and the protein in our bodies? Think of the protein in food like recyclable material. If recyclable plastics are made of polymers and recyclable t-shirts might be made of cotton, recyclable protein is made up of amino acids, the building blocks of protein. Humans need 21 different amino acids, and these little units combine to form all kinds of proteins in the body. Take titin, for example. It's the largest known human protein, with over 30,000 amino acids strung together, and it helps to make our muscles more elastic. When we eat protein, our body breaks it down into amino acids, which then travel to the cells, that are basically little recycling plants. Inside the cell, amino acids are sorted and rebuilt into the specific protein that each cell needs. But not all proteins are equally easy for our bodies to recycle. That's why scientists talk about bioavailability, a measure of how efficiently our bodies can access the amino acids in different foods. So protein isn't just something we eat. It's a vital part of what makes us who we are. To understand proteins better, it's useful to look at how we first found out about them. Back in the 1830s, scientists were busy trying to figure out what made up the cells of all living beings. Jöns Jacob Berzelius and Gerardus Johannes Mulder, chemists respectively from Sweden and the Netherlands, whose names I'm surely pronouncing wrong, were poking around with everyday foods like milk and eggs. And they stumbled upon something that left them scratching their heads. A large, complex molecule that seemed to be everywhere in living things. In 1838, with a nudge from Berzelius, Mulder decided to name this mystery substance protein, borrowing from the Greek word prota, which means of first importance. For a few decades, our initial understanding of proteins collected dust on bookshelves. But then, in the early 1900s, scientists finally had a tool to actually look at them. They realized that proteins' function depends on their shape, which made the study of their structures both crucial and fascinating. Over the years, scientists created more and more tools, each one better at identifying proteins and their roles. Just this year, a tool called AlphaFold 3, developed by DeepMind, was introduced through a paper in the scientific journal Nature. It can predict the shapes and interactions of proteins with incredible accuracy, and now offers a huge public database of protein structures. The team behind it, like the teams in many of the discoveries we talk about on this podcast, just won a Nobel Prize. The dream of mapping every protein in our bodies, just as we've done with our genes, is more in reach than ever. And mapping is so important because proteins don't work alone. They're like actors in a massive play, each interacting with the others. To understand them, we need to understand their interactions. That's how the field of proteomics was born.
Proteomics refers to a sort of technology for trying to assess and analyze all of the proteins found within the human body, for example, or any organism that you're studying. So the human proteome, for example, is all of the proteins within the human body. Proteomics is the technology for analyzing all of those proteins together.
At first, we thought that by mapping out the genome, like we talk about in episode 1 of this podcast, we'd uncover all the secrets of how the human body works, including what causes disease and what keeps us healthy. But as it turned out, even after decoding the whole genome, a lot was still missing. That's because there's a lot more to health and disease than just our genes. And in the end, it's proteins that bring diseases to life in the body.
Proteins are very dynamic and they're always changing, and they're usually a very good reflection of more precisely where you are right now. Most of the therapeutics and drugs that are on the market are actually targeting proteins because we've discovered a specific protein-related mechanism for that disease, and that's what that therapeutic or drug tries to target and then alter in some way.
Proteins can help medicine in two big ways. They improve diagnosis, and they can improve treatments. Let's start with diagnosis. Proteins act like clues. When we're sick, certain proteins linked to that illness shed and so appear in our blood. Our blood has many tasks. It carries oxygen, nutrients, and our immune system fighters throughout the body. But it also works as a garbage disposal system. For this reason, researchers can sift through it to find these suspicious proteins and figure out what might be going wrong.
One disease with which a lot of people are probably familiar is Alzheimer's disease. There we know that, for example, tau proteins and amyloid beta proteins are involved in the development of dementia and neurodegeneration and Alzheimer's specifically. But there's lots of examples you could give across lots of different diseases, from cardiovascular disease to diabetes to kidney and liver diseases.
And now, to explain it better than I ever could, here's Michael Snyder, a genetics professor, the director of Stanford's University Center for Genomics and Personalized Medicine, and a leader in the fields of functional genomics and proteomics.
And so the power of the proteome, which is measuring all your proteins, or at least as many as possible, is you're measuring many, many of these things. And so if something goes off in your liver or your kidney or what have you, you'll see multiple markers show up in the blood. And by doing these advanced proteomics technologies, you can actually see what's going on. Another classic case from a clinical side is you may have heard of troponin. That's a marker that people will follow for a heart attack. But these days with proteomics, you can follow many, many more markers.
The idea is that proteins could give us a clear snapshot of our health at any given moment. Over the past few decades, scientists have been busy building models and techniques that, from a quick blood test, could reveal our risk of different kinds of diseases, even if we're young and feel perfectly fine. Back to the blood as garbage cleaner idea. The magic here is spotting certain trash proteins way before they cause trouble. These sneaky signs can start showing up years, if not decades, before a diagnosis would come along and surprise us.
Disease is not kind of black and white where you go your whole life not having it, not having any of the sort of preclinical or other symptomatic manifestations of the disease, and then the next day, just all of a sudden, magically, you have the disease. Disease is more like an accumulation over years and possibly even decades, and we see that reflected in the proteins. This is the hope for using this kind of proteomic information, is that we can use it in a preventative fashion where if you can catch this accumulation, this steady sort of degradation over time that leads to disease, if you can catch it early enough, you've caught it at a point where you're able to course correct. Because once you often get to the full-blown disease, it's often difficult to completely reverse and eradicate it. Certainly the shift is from being reactive in terms of our healthcare to being proactive and preventative.
But proteins don't just signal disease. Sometimes they are the disease, clumping together where they shouldn't be, or acting up in unexpected ways. The silver lining is that these proteins can give medicine a roadmap for finding a fix. We might be able to craft personalized drugs that stop the disease or at least calm things down a bit. Personalized drug therapy promises to use data from the DNA and from the proteins of each patient to create treatments that are more effective and cause fewer side effects. For example, a study that uses data from hundreds of thousands of patients from the UK found only 38 proteins linked to different kinds of diseases, like inflammatory bowel disease, coronary artery disease, and schizophrenia. By identifying these proteins, scientists could also pinpoint more than 1,000 compounds that might work as potential treatment for them. But again, with such powerful diagnostics, if you catch the very first whispers of a disease, you might not even need meds just yet. You could try other options first, giving your body a chance to sort things out before going the pharmaceutical route.
The big difference between looking at proteins versus genetics, for example, is that since these are dynamics, we can use it in an iterative way as a sort of feedback mechanism for care. So, for example, when you do a first test, you find that you're high risk for cardiovascular disease. But we know that there's a number of things you can do to reduce your risk of cardiovascular disease. Maybe you and your physician start to put you on some alternate sort of lifestyle behavior modifications. Then 6 months' time, a year from now, you take another test and you can see, have you made progress in the direction where you want to go?
So it's no longer just, exercise more, eat well. It's very, very specific. Eat this because this is what's going on for you. And I think we're only going to get better at that as we go into the future. Nobody wants to learn there's something going off, but it's better to learn it early before you have symptoms. But I think it's up to the person to figure out what information they can best handle and then have it delivered accordingly.
This is the vision. And of all of the revolutions we've covered in this podcast, it's the one that feels like we're still in the middle of the story, with enormous potential and some limitations to overcome. Right now, there are still limits to what we can measure and detect. Out of the 100,000 known proteins in the human body, we've been able to capture maybe 10,000. So there's a lot we don't know. And proteins aren't exactly a quick snapshot. They're more of a moving target. They're like stars in distant galaxies. By the time we spot them, they may have already changed and shifted around. Studies have shown that in just 7 days, specific proteins can shift wildly based on what we eat or do. Which means that, for now, there's a fair bit of noise in our individual measurements.
I don't think we have a good enough handle on the sort of time dynamics of how these proteins change. I think we're just scratching the surface of understanding just how responsive they are to different behaviors, to different things you're exposed to in your environment, and on what time scale and how reversible that is and modifiable it is. If we can leverage the power of sort of large population data to understand these ranges and values across the population, we can get more precise at figuring out where are those thresholds above which or below which we should be concerned.
Another surprising thing we can see when we track people's proteins is that some proteins seem to change consistently more than others. So looking at proteins is a bit like using one of those face filters that gives you a sneak peek at what you might look like decades from now, based on the fine expression lines that are already present on your young face. Only, instead of a makeover for your selfie, we're getting a look inside our bodies to see how our internal landscapes might age.
And it turns out that everybody's aging differently. So some people, we call them cardio ages. We call this ageotypes. Some people, their heart pathway seems to be shifting. For other people, it's metabolic. For other people, it's immune. And for some people, it's a combination of all of those. So, and you can measure this. And it turns out the information is actionable. So if you see someone's a metabolic ager, you can kind of encourage them to maybe exercise more or lose weight if they're overweight. It depends on exactly what's going on. You can tell, for example, what your heart age is or what your, oxidative stress is and or inflammation from cytokines. These are all proteins. So that if your biological age is older than it should be for heart, well, then maybe definitely exercise more, maybe get a few things checked. Maybe it's fibrosis, and you can pick that up with signatures. And there are specific things you can do for that.
With all these potential advancements in precision medicine, especially in preventative care, when would we even start being patients? The word patient comes from the Latin patiens, which originally meant one who suffers. But in this new landscape, patients could possibly not suffer at all. They'd be proactive individuals taking charge of their health while still feeling great, ready to tackle matters head-on, rather than waiting for something to go wrong.
And if it were low cost, you could do at least once a month, and you'd catch things when they're shifting off pretty early. And I think that would be very powerful.
And of course, not everyone has to do that. Some might prefer to live happily without thinking about potential problems until they arise. And that's perfectly fine. But the real question is, will these advancements truly be accessible to anyone in the population?
So I think there's an equal responsibility for providers and healthcare settings to take advantage of this and to proactively be testing for things that an individual person just doesn't know about. It shouldn't just be that the benefits of preventative medicine or personalized medicine are only experienced by those who know enough to get the very specific tests that they need to, and then have the resources to then make whatever changes to their life they need to do. We need to make sure that we have safety net and responsibility for the sort of correct factors and aspects of society that pull in and help all of those people who don't know about that, and who help those people make whatever modifications and changes to their lives they need, regardless of whether they have access to the resources to just do it on their own.
And so I do think we need routine healthy profiling. Now, the problem is that our healthcare system, at least in the US, is broken. We're not set up for that. Nobody pays to keep you healthy, and we don't have the financial incentives aligned. So we need to shift that. Meaning, I think you should get paid if you wear a smartwatch or get your genome sequence or get a multi-omics profile, still kind of researchy, but you get the idea. Some more deep data profiles will get checked up more regularly. I think we are practicing sick care, not healthcare, and we would do a lot better as a society if we were practicing true healthcare, where you're monitoring people while they're healthy and catch disease before symptoms come on.
The thing is, for a government or public healthcare system, paying for someone's treatment is far more expensive than investing in preventative measures. So it would make sense to steer towards a more personalized preventative system, right? The catch is we're stuck in reactive mode, always playing catch-up, shifting gears to prioritize prevention while still taking care of those who need treatments, who are multiplying thanks to longer lifespans but no longer health spans, would require a big, bold investment in public healthcare. Instead, we're seeing cuts everywhere that feel like trimming the sails of a ship that already risks sinking. Timely access to affordable, quality preventative and curative care is supposed to be a key part of the European pillar of social rights. But in many European countries, public healthcare systems are underfunded. We seem a long way from a system where we're ready to spend money on expensive and deep profiling checkups just for the sake of checking, without any urgent warnings.
The way I see it now is we're like an automobile industry without a dashboard. We don't really do continuous health monitoring, but we could. You don't imagine driving a car without, its various sensors on it, your gas gauge, speedometer, and all the engine lights, all that stuff. That stuff's all running in the background and displays on your dashboard the health of your car. And so we need the same thing for humans. At least in the US, a physician spends about 15 minutes with you when you do a visit. And so that means they really don't have much time, and it's not their fault, it's just the nature of the system. So the best person to take care of your health is you.
There's a lot to unpack here, but a pressing question looms on the horizon. Are we about to witness yet another disparity where the wealthy manage to avoid diseases altogether while those with lower incomes are left to confront the harsh realities of illnesses and have to pay for it? The European Union agenda aims to make personalised medicine more accessible across its member states, trying to address these disparities in healthcare access and quality. Within member states, we will need to be mindful of our priorities, ensuring that the benefits of these advancements reach everyone, not just a select few. That little tangent was bound to happen, but let's steer back to proteins. What's next? What's in sight? As we mentioned at the start of this episode, no protein operates in a vacuum. Everything in our body is part of this big, interconnected mess of simultaneous happenings. So the real power won't lie in the proteome alone, but in how all of these new layers of understanding of our bodies, the ones we've discussed in each episode of this podcast, interact with one another. I like to think of the complete genome, the proteome, and the microbiome as distinct maps of the same terrain. You've got one map that shows altitudes and ocean depths, another one that lays out the roads and highways, a third that gives you real-time traffic updates. To really grasp what a place looks like without stepping foot in it, you have to overlay these maps, letting them reveal the intricate tapestry that could explain a lot more than what we've been able to so far.
The dream, what we would love to be able to do is take genes that are discovered from genetic research, for example, and then use protein information to look at the functional consequences of that gene. Does that gene lead to changes in the protein, which is then what's causing the disease? That would be the chain that we would love to be able to establish in science.
In my ideal world, we'd have, deep profiles for essentially all human diseases so that you would routinely get measured. So we'd look maybe at several hundred proteins and say, aha, this set seems to be off. You might have this, go do some follow-up tests and get it checked out. I don't think it will become quite as routine, at least not with these in-depth profiles, because it's kind of pricey. And most physicians wouldn't know what to do with the information anyway. So these projects are really research projects. They're intended to try and make discovery of very valuable markers. And then the hope would be you'd take a set of these markers, and then you would actually develop a clinical health test or illness test.
We shall see. The future looks like an incredibly fascinating puzzle. Complicated, promising, and hopefully within our grasp. This wraps up the first season of The Science of You. I hope you've enjoyed this journey as much as I have, and I'm excited to whisper many more exciting developments into this mic soon. The Science of You is a Cora Media podcast, supported by the European Partnership for Personalised Medicine, coordinated by Fondazione Toscana Life Sciences, and co-funded by the European Union's Horizon Europe Research and Innovation Programme. The podcast is written and hosted by me, Silvia Lazzaris, with editorial support from Simone Clemente and editorial curation by Graziano Nani. Sound and music supervision is by Luca Micheli. Post-production and editing are done by Andrea Girelli. The sound engineer is Luca Possi. The project manager is Silvia Arcidiacono Mutti. The producer is Martina Conte, with post-production coordination by Matteo Scelsa.